So the next speaker I'd like to introduce is Dr. Melissa Haendel, and she's currently at the Oregon Health Sciences University, and her talk will be about making connections between human and animal phenotypes. Welcome.
Thanks very much. So I'm going to switch gears a little bit and talk about an informatics project called the Monarch Initiative specifically, but more generally, how do we go about better using the data that are collected from many of the precision modeling wet lab types of experiments that many of you all are deeply involved with? So I work a lot in the rare disease field where we really think about how we can better leverage the data that's out there for disease diagnostics. And so if you look at what the current prevailing pipelines, I lost my title there, involve, they largely focus on use of the genomic data, and I'll get to that in a minute, but basically, trying to find the shooting gun that's the variant of significance in a family to help diagnose a rare disease patient. And that's done by, a variety of different algorithms, but also by comparing with public data sources. The problem is that we don't really actually use much in the way of the other types of data that might exist about the patient. So really understanding more deeply how do we compute over clinical phenotypes, omics phenotypes, all the socioeconomic factors that affect clinical presentation, environmental exposure data, and many other types of data about the individual patient. And then similarly on the public data side, what's the way in which we define disease itself, the phenotype definitions, population frequencies, correlations with disease for these features, and then population-based risk statistics, all the GIS and exposure data that we might want, and the list goes on. And so essentially, we have this large suite of underutilized data. So some of the work that our group is doing is really trying to sort of scratch the surface on how do we actually leverage more of these data from the patient side as well as on the public population and cohort side. So, of course, one of the reasons we're here today is because more species equals more knowledge. So we've been spending quite a number of years to try to determine how do we actually better use the model organism data that's published in the literature and in model organism databases and many other public resources to help inform disease diagnostics. So here we actually have 19,000 genes that were presented in the EXAC database when it was published in 2016. And what we did for here is we actually looked at, if you look at those data, approximately half of those genes have a causal mutation associated with them. So there's a clinical disease or negative phenotype associated with approximately half of those genes. So that doesn't bode well for the other half. So in a diagnostic setting, that is. So if we actually take the orthologs of those 19,000 genes and we look at the orthologs of the top 4 most widely used model organisms here, so the zebrafish, the mouse, the fruit fly, and the worm, we had 14,000 of those genes actually had causal mutations associated with them, so where we had a mutation in the gene and we could see a negative phenotypic effect. So if you combine those, we actually get 89% coverage of this gene set. So it's really a lot of phenotype data coming from the model organisms that could be used to help inform disease diagnostics. So the problem, though, is that we live in a world where there's the phenotypic chasm of despair. So the clinical data, I don't know if I have a pointer or not. How do I point? I'm going to probably do something bad here. Oh, there we go. So, over here on the clinical side, we might have things like wearables. We have lots of specimen data, path labs, all kinds of different clinical instruments to try to, get information from patients. Many clinical descriptions of diseases and syndromes, medical imaging data, the messiness of EHR data and all of the complications that, arise in that context because we don't actually describe the patient as a biological subject in the EHR. We describe them as a billing subject or quality of care subject. And so how do we actually go about describing the patient in the same way that we might describe a model organism? And so then over here, you know, in the model organism world, we really do a very good job of describing our biological subjects in terms of their anatomy, their morphology, gene expression, metabolomics, all kinds of molecular modeling, cell models, et cetera. And so the list goes on. And the problem is that these data over here just are not, in a computable fashion, really very relatable to those data over there. So, one of the reasons, and this is just one of many, but the one that we're really trying to tackle is that the different communities use different language to describe phenotypic features. So here we have a clinician that might describe a phenotypic feature in a patient as palmoplantar hyperkeratosis, where the patient might describe and go to their clinician and say, I have this really weird, you know, thick hand skin. And a mouse biologist who might see a similar phenotype would say that the mouse has ulcerated paws. So how in the world are we going to actually reconcile these very similar phenotypes across these different communities? And the same thing is true even within a model organism community or within a clinical community, that these language barriers really become very problematic. And the situation is even worse when we think about this problem globally for rare disease. So at the end of the day, we have lots of standards to choose from. Over here, we might have the human phenotype ontology. We've heard a lot about the IMPC, which uses the mouse phenotype ontology. But the reality is that the situation actually looks a lot more like this. And there's literally hundreds, if not thousands, of vocabularies and standards used to describe phenotypic features in the same species or in very many species. And so we have, you know, what's really a semantic mess out there in terms of these standards. So how do we actually make sense of this data when everybody is standardizing their data against a different standard? So what we basically have tried to do is to try to figure out how do we tell the computer what palmoplantar hyperkeratosis really means. So to do that, we basically define an underlying logic structure that allows us to describe that particular phenotype, palmoplantar hyperkeratosis, in the human patient in a species-neutral way. So over here we have an ontology, a vocabulary of qualities like increased. We have the gene ontology, which describes biological functions in a species-neutral way. Here are keratinization. We have the Uberon anatomy ontology that describes mostly vertebrate ontology, but it's a cross-species ontology that can be subsetted for individual species. So here, the stratum corneum layer of the skin that's located in the autopod. And so we create essentially a logic structure that combines these terms from a species-neutral ontology, so that when we see that mouse term, ulcerated paws, that has the same or similar related logically description, we can actually know that these two phenotypic concepts are related. So one of the key elements in using this type of approach is actually a new way of describing clinical phenotypes. So this is the human phenotype ontology, and people often ask us, you know, why do we need yet another clinical phenotype ontology? Aren't you contributing to that problem of the 14th standard? And the answer is, well, actually, we think that we do. And one of the reasons that we do is that these so here we would represent a patient or a disease as having a set of nodes in a graph. So here this patient or disease might have the phenotype of hyposmia, deeply set eyes, motor neuron atrophy, where essentially the collection of nodes in the graph that represents that patient is sort of a phenoprint. It's the sort of characteristic phenotypic profile of that patient, but it's represented in a computable way. What makes the human phenotype ontology different are two things. One is that each one of these nodes in the graph is represented in that logically computable manner that I described earlier. So here, hyposmia is described logically in terms of the sensory perception of smell, which has 34,000 annotations in 22 species. So all of a sudden, there's an enormous amount of data that can be associated with simply just that single term. And if we think about the patient as a collection of these terms in the graph, then we really can combine data from very many different sources. The second thing that differentiates the human phenotype ontology from other clinical vocabularies is the extensive set of annotations that we have created to represent known human diseases, whether they have a molecular basis, known or not. And so here we have actually up to about 14,000 terms now, with over 127 rare disease annotations done in collaboration with Orphanet, and 136,000 common disease annotations. So essentially creating one of these profiles based upon evidence from the literature, from many different database resources such as OMIM, Orphanet, and many other sources, can be the sort of gold standard corpus against which we can build algorithms for diagnostic use. So we're basically using that logic-based approach to integrate data across many different genotype, phenotype sources that are both clinical as well as model organisms. So up here we have many different types of genotypic information, so it could be genes or variants. We have many different types of associations, only a few of which are shown here, and many different types of phenotypic data or disease data. And so this is just a few of the sources. They each use their own favorite flavor of ontology, some of which we contribute to. And we have a variety of those sort of species neutral or sort of unifying ontologies that then allow us to bring these data together. So over here we have many different disease resources, MedGen, that's used for submission to ClinVar, the medical subject headings, the disease ontology, OMIM, the Orphanet ontology, elements of morphology, and there's many others. So we have a unified disease vocabulary that actually harmonizes all of these into one nosology. Similarly, we do the same thing for phenotypes, Uberon I mentioned earlier for anatomy, and this genotype ontology, which actually is one of our biggest challenges because each one of these sources associates different aspects of the genotype to the phenotype that's being recorded. So in some databases, we'll get a disease being associated with the gene, and another one, it'll be to a variant. Sometimes it's a haplotype, sometimes it's a region of the genome. And so how do we crosswalk and combine data when the genotype-phenotype associations and the evidence for them is actually associated with different components of the genotype? So when we combine all of these data, we end up with over 2.2 million genotype-phenotype associations, and 91% of those require 2 or more data sources to create the association. So this is actually a really powerful point because it means that data sharing and data sharing well can actually lead to new associations that might be used computationally for diagnostic use. So how does the diagnostic use actually work? So here we have a rare disease patient, undiagnosed here. Patient A has a set of those phenotype terms, so hypothermia, seizures, optic atrophy, and we basically have a fuzzy matching algorithm that will look against those known diseases, so over here disease D, which has a mutation in gene D, to find the most closely related graph. And in cases where we don't find one that's a known disease, we go to the model data. And so over here, our patient with, I'm having a hard time reading upside down, generalized amyotrophy might be related to dystrophic muscle over here with a term in common, abnormality of the muscle morphology. And so you can see that then that the ortholog of gene M might be a candidate for patient A's disease. The other thing that this allows us to do is it says, well, this model organism researcher over here with this mouse is not only are we potentially matching this mouse, but we're matching against the expert here. So this expert may be an expert in phenotyping these types of phenotypes and could be a good collaborator for this clinician over here for where they may have variants that need to be functionally validated like we heard about this morning. So this approach is combined together with more traditional variant prioritization methods in a tool called Exome Iser, which Damian Smedley is going to talk about more tomorrow, but just to give you a quick introduction. So basically you take the whole exome, you remove off-target common variants, you apply Mendelian filters, you have your family, you get a variant score, and you can combine that with this phenotype score as well as with protein-protein interaction data or other kinds of data like I showed you to help sort of have a guilt by association type of approach. And then that gives us a final score of candidates. So in collaboration with the Undiagnosed Disease Program, we've applied this technology and have improved the diagnostic rate between 10 and 20% for the Undiagnosed Disease Program. And so this is just one example where we were able to diagnose a family with a heterozygous mutation in the STIM1 gene based on that type of fuzzy phenotype matching to a mouse that was available publicly and described very well in the Mouse Genome Informatics database, where there was no correlate in OMIM, dbSNP, or ClinVar. And tomorrow, Damien's going to tell you a little bit more about sort of work in progress to use this tool across the 100,000 Genomes project. The other thing that this allows us to do is it allows us to matchmake patients across the globe. So when we have an undiagnosed patient, we're really trying hard to determine how is it that we can find that second family and thereby validate the candidate variant that the patient may have. And so in this context, we have a clinical site that has what we call a matchmaker exchange site set up, and this is part of the Global Alliance for Genomics and Health. And in this site, they describe in a sort of structured way the phenotypic features of the patient in the same way as I showed earlier, and then the candidate variants that patient may have. And then that matchmaker exchange then queries other clinical sites around the world, as well as open databases like the Monarch Initiative, which combines all of the open data to make sure that candidate diseases as well as candidate models are available in order to help inform the diagnosis, as well as actually assisting in potentially finding collaborators. So then I just wanted to talk really briefly about some of the challenges in sort of modeling disease from a computational perspective. So traditionally, there's been a lot of efforts to just say, well, if you have, oops, let me go back. If you have, holoprosencephaly as a disease caused by mutation in sonic hedgehog, we'll just look at the orthologs of that gene and make mutations, and maybe we're making a knockout, or maybe we're making a humanized mouse in this case, and then that's thereby a model. And obviously this has huge benefit, but also huge challenges. We also have a lot of data that comes in where people have just asserted that a given model is a model of a disease. And in this case, what they're really saying is that it's a proxy for the fact that it somehow recapitulates phenotypic aspects of the disease. So my premise is, why don't we just use the phenotype data itself to help us determine whether or not it's a good model of the disease? And so this is some tools that we've developed to actually display the phenotypic similarity between known diseases and known models to try to find the sort of nuances of which aspects of the phenotype are actually being recapitulated. And I think the idea should be that, and this came up in some of the earlier talks as well, that we really fundamentally don't necessarily, shouldn't expect to recapitulate every aspect of the phenotype in a model. It's a different organism. We're going to learn different things from different organisms. What we really want to look at is the underlying mechanism and pathology. And those are the things that, if we're trying to investigate, we want to sort of parse out and find the best model. So Jennifer really gave me a great lead-in because I think that we need to really be thinking more carefully about which organism is best to model the particulars of a given disease. And so I just put some examples up from some of the work we've been doing to actually sort of inventory all of the models of disease that have been published in the literature. So, for example, naked mole rats don't get cancer. We just heard about marsupials having very rapid craniofacial and jaw morphogenesis. The pig eye is actually the one that most closely resembles human eyes. We have an example here for C. elegans. The C. elegans ortholog knockdown leads to increased DNA cross-linking, which is probably one of the best models for that part of the phenotype for Fanconi anemia. So there's just many, many examples of where we have fundamental phenotypic similarity for individual phenotypic elements in a human disease in what are thought of as non-traditional model species. So now we're trying to develop new informatics strategies to help inventory and assess the model suitability for modeling specific sets of phenotypes, and then also just to better justify our choices. So for IUCOOK, but also for ethical reasons, how can we actually justify the organisms that we're choosing and not just choose the ones that we happen to have in our basement? Not to say that there aren't some extremely good reasons for using the ones that we have familiarity and resources to help support, but we should do collectively as a community a better job of understanding what might be the best model to choose. And this is just an illustration of how we've actually, as we take an informatics approach to this, we just learn very different things across the different species. And so this is an illustration of where we're getting to with trying to create tools for choosing the best model. So, and then finally, I just wanted to mention another effort that's part of the Global Alliance for Genomics and Health, which is a phenotype exchange format. So, we have lots of standards for exchanging genomic information and sequence data, but we really don't have a very good mechanism for exchanging phenotypic information, which is fundamentally one of the problems, is that it gets locked up in the literature, whether it's patient data, patient descriptions of cases or cohorts, or model organism data, requires an enormous amount on part of curation and then representation in public databases that then are all not really quite interoperable and take a lot of expert effort to make interoperable. So this is a community effort to try to, kind of structure at least some proxy of the components that are present in the literature for describing phenotypes in a way that you can at least use it computationally to query and find similarities within and across species. So just to thank all the enormously smart people that actually do all of the work. So in my group, Matt Brush and Kent Shefchek do most of the data modeling and data integration work. Julie does all of the beautiful illustrations. Damien Smedley is here today, and he leads efforts on the Exome Iser tool. Peter Robinson leads efforts on the human phenotype ontology and the algorithms. Chris Mungle is an ontology engineer who has really led the development of the gene ontology underlying technologies and a lot of the ontological modeling and engineering. Let's see, who am I missing that I need to get to? That's probably good for this talk. And then last but not least, I also wanted to thank all of the data providers. So without these data sources making their data publicly available, we would never be able to create these types of informatics methods. So we thank all of our sources as well as our funding agency, and we're funded by the NIH Office of Director through the Office of Research Infrastructure Programs. So thanks very much.
Thank you, Dr. Haendel.